So hello everyone, my name is Anne Ibbotson. My background is in veterinary science, but this year I recently commenced a PhD in the Conservation Science Research Group at the University of Newcastle. The Australian Wildlife Society is kindly funding my research project to see whether stress and reproductive hormones can be integrated with ecological measures to improve conservation decisions for the Little John's tree frog, which is this beautiful tree frog you can see here. So today I'll be speaking about this project and my PhD more broadly, which includes measures of immune function and extends to the green and golden bell frog. Frogs are fighting their own pandemic, the fungal skin infection chytridiomycosis, or chytrid for short. It's devastating populations, and we've added human-driven threats such as habitat loss and fragmentation and climate change. The situation is critical. Over 40% of amphibian species are threatened, and this includes the Little John's tree frog and the green and golden bell frog. Unfortunately, further declines are occurring in many species despite conservation efforts, highlighting that new approaches are needed both to understand the impacts of these threats on health and to also monitor and inform conservation efforts. My project involves developing minimally invasive biomarkers of amphibian stress, reproduction, and immune function to inform environmental management decisions. We know chronic stress has negative impacts on health, and it's important to understand the interplay between stress, reproduction, immune function, and disease, as it has major implications for population health and ultimately species survival. Conservation strategies are in place for my species, including breeding programs and initiatives to create artificial ponds and connect habitats, but biomarkers provide a platform to build on this. I plan to measure the main stress hormone, corticosterone, the reproductive hormones, estrogen, testosterone, and progesterone, and a novel marker of immune activation, neopterin, in urine using mass spectrometry, which is considered the gold standard of hormone analysis in people. I'll then correlate these results with reproductive output and status by measuring egg size based on ultrasound, which are these little white things that you can see on the screen, and sperm quality in males. I'll then also correlate those results with genetic diversity as a measure of inbreeding, chytrid status, and ecological measures, which should allow us to compare populations and also look at the relationship between chytrid and reproduction and stress levels in these frogs. Another aspect of my project involves collecting skin swabs to perform immune gene expression studies using qPCR, which is a similar technology that you have in the COVID tests. This will improve our understanding on the frog's immune response to chytrid infection, and we can also look at the immune response to temperature variation as a measure or as a mimic of climate change. These minimally invasive methods are vital in endangered species where every individual counts and are critical to improving animal welfare in wildlife research. Biomarkers, together with ecological measures, can help us detect stresses in the environment, gauge the success of artificial ponds and optimize conditions in our breeding programs, and can also improve our understanding of impacts of climate change as we monitor biomarker response to temperature variation. To conclude, biomarkers enable us to view populations through the lens of individual animal health and have the potential to be integrated into decision-making for conservation actions for threatened species. Thank you very much for listening today and giving me this opportunity to share my research. And also thank you to the amazing team of people that I work with.